in collaboration with Digistain and Demasa. We're honored to have our two distinguished speakers, Professor Charles Coombes and Dr. Himmel Amrania, who will share their expertise and groundbreaking research on this innovative technology. Please share any questions throughout the session in the chat. We will address this in the Q&A session after our speakers have given you some insights. And then, yes, thank you for your dedication to advancing healthcare and improving patient outcomes. We hope you find this webinar both informative and inspiring. Professor Charles, handing over to you.
Thanks, Terry-Leigh. Well, it's a real pleasure to be able to talk about this. This has been the subject of research at Imperial College in London for the past 12 to 15 years. And it came about through, I guess, my recognition that Chris Phillips and Himmel Amrania had a system which could very rapidly derive prognosis in cancer. Next slide, please. So, as we all know, I don't need to tell this audience that the three main types of breast cancer are hormone responsive, which are estrogen receptor and progesterone receptor containing, and that constitutes the vast majority of cancers, around 70%. And then there is a 25% of patients have HER2 positive breast cancer, and the remaining are triple negative. Next slide, please. So the key question, I guess, that faces us all as oncologists is to try to derive a prognostication for our patients. We know that size and lymph node status are important, but it doesn't really tell you a lot about long-term risk. Next slide, please. And so you'll see that the outcome of regional, i.e. patients with lymph node involvement, is much lower than those patients with localized disease, and late recurrence occurring more than 5 years in hormone receptor positive breast cancer is quite common. And when you develop distant metastases in breast cancer, you'll see that the chances of 5-year survival are very low. So we all need to do what we can to prognosticate correctly and give our patients the correct treatment. Next slide, please. So we all know that endocrine therapy is our standard treatment, tamoxifen, and sometimes with an aromatase inhibitor, or ovarian suppression in premenopausal, or tamoxifen, or adding with an aromatase inhibitor with premenopausal, and in postmenopausal women, as you know, most women get aromatase inhibitors at the moment. But the real question that we all have to find an answer to is which patients need intensive treatment, which patients, for example, with premenopausal breast cancer, need the combination with an aromatase inhibitor. We know that this causes quite severe side effects. And of course, which patients need chemotherapy in addition. Next slide, please. So I think we can just go on to the next slide. Of course, this is about side effects, which I mentioned and which we all know about with chemotherapy. So what are the current methods? So at the moment, I guess most of us use Oncotype DX. This is the 21 gene expression profiling test, and normally in patients who fall in this middle range of patients that, most of the patients we know either need just endocrine therapy or endocrine therapy and more intensive endocrine therapy or chemotherapy. But it's the quite large group of patients where we are just not certain. And that's where Oncotype DX comes in, which is this ability to predict a 10-year distant recurrence risk, and I guess a risk, a figure of more than 26% indicates chemotherapy. And most of us, I think, use this test at the present time, but there are problems. So there's limited data for patients with node-positive disease, and we don't have much data beyond 10 years. And we'll go into the time taken to get the results back in another slide. Now, MammaPrint is a 70 gene expression profiling test, and it again guides adjuvant chemotherapy decisions, especially in high-risk patients, but has less specificity compared with other tests regarding hormone-positive subtype. And again, we've got limited data on beyond 10 years. EndoPredict is less widely used. It's got limited data on its ability to predict chemotherapy benefit. And then there's the PAM50 gene expression profiling test, which again requires specialized equipment, and it's less data. We have less data on its ability to predict the need for chemotherapy. So in summary, I think this is on the next slide. So in summary, there are several sort of limitations in the gene expression profiling tests. Obviously, cost is one factor, and I think someone else will be covering this, but the main problem from me as an oncologist, I guess, is the turnaround time and the also the lack of direct comparisons. You know, most of us are kind of assuming that these tests are similar, but in fact, they haven't been compared. We don't know. And the turnaround time for me is the major problem. Patients are very anxious in this 2 to 3 week period that's needed to get these results back. And it was really that I guess was the main motivation to work with Chris and Hemel to try to improve on this situation. Next slide, please. We could go on. Next slide. So basically, just to look at these again, and the variability issue is summarized here. That, for example, John Bartlett published several years ago, 39% of patients had discordant results across Oncotype DX, MammaPrint, Prosigna, and IHC4. And in addition, Mitch Dowsett published some years before that the agreement between Oncotype and Prosigna in low risk categorization was only 59%. Next slide, please. I think the other problem is that there is some degree of variation in treatment recommendations. So that a study which was published a few years ago, it was a retrospective analysis of 145 breast cancer patients, showed kind of a third of oncologists disagreed on whether chemotherapy recommendations for Oncotype DX were correct. So there is a sort of vagueness, I think, and a lack of general agreement by us oncologists about the significance of certain numbers on the Oncotype DX. Next slide, please. So I've mentioned this. This is was my main motivation, is the treatment time delay. I don't need to keep going on about this, but the degree of anxiety is a problem. Next slide, please. And then, of course, this issue, which I mentioned, the limited applicability to certain patient populations, particularly in the case of Oncotype DX in node-positive women, and the New England Journal paper published only 3 years ago showed that for premenopausal women, the patients with 1 to 3 nodes seem to benefit from chemotherapy regardless of the recurrence score. So we are in a situation where a lot of these tests are not very satisfactory for subsets of patients. Next slide, please. And then in terms of other risk factors and other treatments, and the other prognostic factors which are constantly being unearthed. There are many studies. This particular one, the New England Journal, found that clinical risk factors provided additional prognostic information beyond the Oncotype DX recurrence score. So suggesting a need for a slightly more comprehensive risk assessment tool. Next slide. And then, of course, the long-term outcome prediction. This was the paper by Pan et al., again in the New England Journal, with more than 60,000 women about the long-term recurrence risk. And as I've said, a lot of these tests are limited to around 10 years. Next slide, please. So in summary, I think it's quite clear that risk stratification is really important in deciding what adjuvant therapy is appropriate for each patient, bearing in mind that quality of life is very important. Secondly, current methods are time-consuming and to some extent subjective, and for these reasons, I think there is room for a better, more rapid risk stratification method to enhance patient care. Next slide, please. So Digistain, we in fact only published the first paper 6 years ago, but the final analysis with a long-term follow-up of many patients was published in Breast Cancer Research and Treatment only this year. It does provide a direct measure of elevated levels of DNA, and we know that aneuploidy is something which is common in aggressive breast cancer, but that there is no objective way of assessing aneuploidy. And I think because this form of spectrophotometry gives a different signal depending on the amount of aneuploidy, you've got a proper readout of the degree of elevation of DNA, which you obviously see in aneuploidy. Secondly, it delivers a result in 15 minutes, which is, I think, quite outstanding. And finally, in terms of the negative predictive value, it has a very similar NPV to market-leading tumor risk profiling tests. Next slide, please. So how does this work? Very simply, there are 5 steps. First of all, you get tissue slide, very similar to the way that is being done with conventional pathology. The difference is that you don't need immunostaining, and you don't need an H&E preparation. You don't need technicians to do this sort of thing. You put the slide directly into the infrared spectrometer, and this generates a very complex signature for each patient. More than 10,000 data points are amassed, and the apparatus processes this data through AI, analyzes it very rapidly, and then predicts the recurrence. And in this case, it's just an illustration. There's a low risk of recurrence, a less than 10% in this case, and you get a percentage chance of overall recurrence. So that is a simple depiction of the methodology. I'm sure that Hemel and the team, the Digistain team, will give you more scientific explanation if you need it. And I think the final slide is really just the references. I think you may have that on the files. Yes, you do. But I just want to thank Chris Phillips and Hemel for the time that they've spent developing this system over the last 15 years. I think they've done an amazing job. And I'm so pleased that they have now managed to set up a company that can get it distributed and used more widely. Thank you very much.
Thank you very much, Professor Coombes. I am happy to hand over to Dr. Himal Amrania, who will take you into a deeper discussion on the profiling solution. Dr. Himal?
Hello and good evening, everybody. So a massive thank you to Charles. He has paved the way for us really to get Digistain to market, and indeed, as many of you are aware, he's one of the leading voices in breast cancer today. So although he might be annoyed at me for saying that, I think it's very true, and it's we're immensely grateful for having him on board. So to echo what Charles said, Digistain was the innovation behind Digistain was created in response to a pressing clinical need for a more cost-effective and streamlined solution for triaging patients by risk, so that they can choose, so that physicians can choose the right adjuvant therapy. Next slide, please. So today in South Africa, we have officially reported over 10,000 cases of new breast cancer cases annually each year. Although the real figure is probably a bit more. It certainly is the most common cancer for South African women, and is indeed a national priority in South Africa. Now, what's happening in South Africa because of reimbursement challenges is that very often the first line of treatment, or first line of defense for cancer patients is chemotherapy, which results in all kinds of undesirable sequelae and side effects. And the reason for this is because cancer therapy is very often the biggest cost center to any healthcare system. So here in the UK, for example, although Oncotype is reimbursed by the national healthcare system, by the state, and that means that we ship our cases abroad to the US, California, for analysis to provide treatment decision-making data or insights to our physicians. Now, in places like South Africa, where most genomic tests would not be covered under national medical insurance, physicians are left to use the standard parameters like tumor size, lymph node status, and grade to make adjuvant therapy decisions. And as Charles has shared already, that acting with that information alone, it's fairly limited information. Next slide, please. So I'd like to take us all back 20 years, around 2003, 2004, where we know that the subjectivity in grading, with all other factors being equal, just the subjectivity in grading that leads to an over-treatment of chemotherapy. And indeed, in 2003, 2004, with the advent of genomic tests like Oncotype or MammaPrint, genomics was heralded as the solution to this subjectivity problem. But as Charles did with Bartlett et al., the publication that they shared, there was less than 40, it was discovered that less than 40% of breast cancer tumors are classified similarly by these popular genomic tests. And indeed, in one publication, Luamato et al., Breast Cancer Research and Treatment 2011, it was found 30% of Mammaprint's high-risk cases were reclassified as low risk by Oncotype DX. And this is really just echoing what Charles said, in that there is this idea that genomics eliminates all subjectivity in risk assessment is not so accurate, because when we study the longitudinal long-term data, it turns out that that's not the case at all. And it makes sense, because in this vast parameter space that is genomics, we have a limitless number of genes to analyze. And you've got 70 genes with Mammaprint, 21 genes with Oncotype, 50 genes with Prosigna. Which ones do you choose to give you the information, to give you the treatment decision-making insights? And then furthermore, one extra thing I'd like to add here is that because of this subjectivity around the specific genes that you might want to analyze, in JAMA Oncology 2021, it was published that in the Taylor-X trial, it was the Black and Asian patients who were significantly underscored by Oncotype, suggesting that the actual negative predictive value, specifically for the Black or Asian subgroup of patients, is probably a little bit lower. Next slide, please. So as Charles shared, what with Digistain is a unique solution in that it is pseudogenomic or agenomic in a sense, because rather than relying on any specific genes or mutations, it measures elevated DNA levels that are the result of aneuploidy in cancer cells. Now, aneuploidy or chromosomal instability has been established as a prognostic marker in and of itself for many, many years. And this can be measured. Chromosomal instability in healthy cells varies by as little as 1%, and conversely 20%, up to 20% for cancer cells. So aneuploidy generally as a whole is a hallmark of all solid tumors, or most solid tumors at least. Next slide, please. So what we created with Digistain was a way to measure the subtle biochemical changes in the formalin fixed tissue. So we're not changing the sample format at all. We're using the same kind of sample, the same sample format that's commonly used around the world, formalin fixed tissue, and is therefore readily accessible. And then we're measuring, we're taking a unique spectral signature from the tumor to determine the level of DNA, or the elevated level of DNA in that tumor block. And we do this using spectroscopy. It's a very well-established, validated analytical technique in engineering sciences. And the result is a precise and objective evaluation of risk, because unlike a lot of the solutions that Charles shared with you that are commonly used today, which rely generally on RNA yield, or Oncotype, for example, is a PCR based, uses a PCR based methodology, which requires the isolation of RNA initially by digesting the tissue in some exotic enzymes. There are some challenges associated with this. One of them is tissue accessibility. So with some of those genomic tests, they require a minimum of 30% tumor tissue in a in a tumor slide. This is often not possible with very small tumors or with tumors where the tumor cells are not so prolific or present. So what we do there is we have a pathologist to mark the slide to tell us which part of the tissue to sample, and we follow a very robust validated protocol to identify the area of highest grade cellularity and highest grade carcinoma, and then we sample only that very small region, and that gives us a very specific indicator of risk. Next slide, please. So what we're doing there is we're capturing a unique spectral signature from the tumor, and then the data that is generated, up to 10,000 data points are generated, is sent to a proprietary software platform to analyze using a machine learning based approach and then this measurement is then translated into a personalized score that the oncologist can use to make better treatment decisions. Next slide, please. And the net effect of this is that we're able to deliver these decision-making insights in less than 15 minutes. We are establishing a protocol for hospitals to use Digistain in their own pathology labs. And the great benefit of doing it this way is that unlike a lot of complex protocols, genomic protocols, which require the presence of a clinical scientist, Digistain can be run with lesser trained lab technicians. So here in the NHS, we were evaluated by the National Health Service, and they sent a molecular genomics team into our lab to shadow the whole process of producing a Digistain result. And they were really impressed with the level of training required for their staff to run the protocol. It's a very simplified protocol. It doesn't require any staining, as Charles mentioned, and no RNA transcription or anything like that. The technician places the tissue slide, a simple unstained tissue slide, into the apparatus to produce a test result within minutes. And this is in stark contrast to the current methodology, which requires shipping a sample all the way to California or the Netherlands. I know MammaPrint's quite popular in South Africa, and it's very expensive. It's many thousands of dollars per test, and patients are waiting for many weeks. Next slide, please. So the clinical study, our landmark validation study that was published in Breast Cancer Research and Treatment earlier this year in January, was based on very similar inclusion criteria, patient inclusion criteria, compared with the original validation study for, say, Oncotype or MammaPrint in New England Journal of Medicine 20 years ago. We had 801 patients in the study, and what a lot of physicians are responding to very positively is the robustness of the study because of the very long patient follow-up time, nearly 13 years or 12.7 years. And this data was shared last year at ESMO, at San Antonio, also at ASCO, and it won a prize, in fact, at the American Society of Breast Surgeons meeting for being one of the more effective clinical studies of the year. Next slide, please. So to dig into a little bit more detail, this was a study that was led by Charles Coombes, Ian Ellis, and Carlo Palmieri, all renowned breast physicians here in the UK. And this study included patients less than 70 years old, up to 3 positive lymph nodes, tumor less than 5 centimeters in diameter. Any cases where there was ductal carcinoma in situ, or DCIS cases were excluded from the study. And critically, none of the patients received adjuvant chemotherapy, which makes this much more simple to interpret the study, because you don't have to confound the hazard functions with the understanding that some of these patients received chemotherapy. So all of these patients received adjuvant chemotherapy, and that's what a lot of physicians like about this study. Received only adjuvant endocrine therapy, which is what a lot of physicians like about this study. Next slide, please. So for all three primary outcomes, recurrence-free survival, recurrence, and overall survival, and these were all determined using the STEEP criteria. We showed that with statistical significance, there was good risk stratification between the both high and low risk patient groups, and it was shown that 49% of patients could have safely avoided chemotherapy. Next slide, please. One of the things that is of keen interest to physicians today is risk stratification in premenopausal patients, in younger women. So we looked at subgroups here of this large cohort, and with all of the patients in the premenopausal group less than 50 years old, we saw again good risk stratification with statistical significance p less than 0.05 with a hazard ratio of about 2, and also similarly with the lymph node negative subgroup, very, very robust again with p-value less than 0.05, good risk stratification. Next slide, please. And Charles mentioned about the relatively high negative predictive values we're seeing with this. So when we're looking at any of the subgroups, we're seeing the combined aggregate negative predictive value of the whole population was about 94%, but for 10-year prediction. However, for the 5-year prediction, we're seeing very, very high NPVs around the 98 or 99% mark. And when you look at the more aggregated figure of merit for evaluating diagnostic performance, called the area under the curve, most of these risk profiling tests that Charles introduced us to, they demonstrate AUCs of around 0.55 to 0.75. So for this group of patients, for 10-year prediction, Digistain showed an AUC of 0.75, which is right at the upper end of what is possible today with risk stratification. Next slide, please. And here, the question we're often asked is, how do we compare to other risk stratification tools? Now, I must confess, this is data on file. It's due to be published soon. We performed a multi-center randomized study, retrospective head-to-head study versus Oncotype with follow-up data. And what we saw that for a particular center, this is from Bart's Cancer Hospital, we saw that Digistain's AUC was significantly higher than Oncotype DX's. And for the overall study, data which will be shared very shortly, we saw good concordance between Digistain and Oncotype on the identification of low-risk cases who safely avoided chemotherapy, again verified by the long-term follow-up data. And importantly, for those cases that were non-concordant, where Oncotype predicted low risk and Digistain predicted high risk, of that cohort of patients, or that portion of patients, several of them succumbed to breast cancer in less than 5 years, suggesting that for this particular cohort, or for this study, Digistain had a much higher negative predictive value for the identification of low-risk cases who can safely avoid chemotherapy and remain metastasis-free for 10 years. Next slide, please. Now, one of the key things that we need to investigate in order to achieve mass adoption at scale, particularly in a country like South Africa, is the health economic evaluation. So there was an independent health economic evaluation commissioned by the National Health Service for Digistain. And what they showed was that for where they, first of all, they concluded that the sensitivity and the like-for-like sensitivity and specificity for Digistain was identical to that reported by Oncotype in their NICE evaluation. And then secondly, they found that the savings that could be made in a like-for-like swap, swapping Oncotype for Digistain, the NHS could save £290 million without increasing any patient risk, and that's a key thing for us. The point that we're able to achieve cost savings without increasing any patient risk, and furthermore, to add to that, because of the superior cost effectiveness for Digistain, one can achieve broader eligibility for tumor risk profiling tests, and it was found that 1,266 life years could be saved for lymph node positive patients, for the lymph node positive subgroup. So this was a very robust and independent health economic evaluation performed by the state healthcare service here in the UK, which resulted in very good results. And the health economic model is very similar to that of South Africa. The only difference would be the data inputs, and we're working with Imperial Logistics to provide those data inputs so we can provide a health economic evaluation for South Africa as well. Next slide, please. So moving on, I'm proud to share that Digistain has now been adopted by cancer hospitals around the globe, particularly in areas where that are susceptible to healthcare inequalities. So places in Asia and Latin America have adopted Digistain readily, and even though it wasn't our vision to do so, it has displaced Oncotype in many senses. In the UK, we were evaluated. We went through a rigorous evaluation by both the NHS, a team of oncologists in the NHS, and also Bupa, which is the largest private medical insurer in the UK, before it was commissioned for over half of all private patients in the UK. Next slide, please. This is a technology that is started at Imperial College under the supervision of Charles Coombes and Professor Chris Phillips. Next slide, please. And in doing so, one of the key things that we set out to achieve with this was to avoid being a solution in search of a problem. So unlike a lot of university research that doesn't make it out of the lab because care was not taken to ensure that a clinical need could be addressed without disrupting clinical workflow, we specifically designed the protocol after surveying over, exactly 1,560 breast oncologists around the world. And with their feedback, we were able to design the protocol that would mitigate, what will almost eliminate any chance of workflow disruption in the clinic. It was developed with over $5 million worth of research funding. It's patented, and we've got both UK and European market clearance as well from the MHRA. And as I shared, it's already been deployed internationally. Next slide, please. So here is a typical case study. So this is a study where this patient was from Africa, from Kenya, in fact, and she was flying over to be seen by an oncologist, but she was very nervous about the time for treatment decision making. She was a single mother, and she was looking after a family back home. So it was very important to her that she had a treatment plan organized before she went back home. And the risk of waiting several weeks for a treatment plan wasn't something that she wanted to endure. So she opted for Digistain, and this patient was a node negative patient, grade 2, 2 centimeter tumor with a Nottingham prognostic index of about 3.4. So a very classic risk patient. This patient came back with a low risk score after a Digistain test, and she received endocrine therapy, and she now remains metastasis free after 2.5 years. So she's responded very well to the treatment. Next slide, please. This was another patient. This was a grade 1 tumor, 2.5 centimeters, lymph node positive, and again, a classic intermediate risk patient. This patient came back as high risk with a 14% chance of recurrence, which would classically be intermediate risk for oncotype. But this patient received FEC therapy after Digistain and is now responding well to treatment after 2.5 years. So there's one more anecdotal case I'd like to mention, and that is in the study of oncotype versus Digistain. There's one particular case that was a grade 3 tumor. It was a very small tumor, and lymph node negative, perimenopausal patient around 57 years old, maybe postmenopausal, and she received an oncotype test, and it came back with a low risk score. Digistain came back with a high risk score, and this patient comes with breast cancer therapy apart from breast cancer. Even though the N equals 1, I think. Well, maybe we run out of slides. Okay. With that said, I'd love to take your questions, please. Oh, here we are. So yes, a huge thank you to and some acknowledgements, please, to the clinical team that led to the development of Digistain. Sir Professor Nicholas Wright, he's the ex-president of the Great Britain Pathological Society, and also Darius Francescati, a breast oncosurgeon, and was actually he was responsible for launching MammaPrint in the US. He's now working exclusively with Digistain, and also Charles Coombes, a very celebrated breast cancer oncologist, to have joined the clinical board. Next slide, please. And we've also partnered with Emad Raka as well, who's a very well-known breast pathologist in the UK and has recently moved out of the UK. So we developed the Digistain with Charles Coombes, Sir Nick Wright, and Professor Chris Phillips, and then validated it with Ian Ellis and Emad Raka in Nottingham. Next slide, please. And final acknowledgements for the funders to have supported the study, the research, which include the NHS, the NIHR, and Innovate UK, as well as Royal Society and Harvard. Harvard have invested in Digistain, and so has Y Combinator. We've been lucky enough to have received a number of accolades from organizations such as the Royal Society and the Institute of Physics Innovation Award as well for the innovation. Thank you very much. Any questions? Oh, and one final point I'd like to add here is because Digistain takes a very elegant approach in measuring the elevated levels of DNA, rather than trying to rely on very upstream or biologically upstream genomic markers or mutations that may or may not result in tumor regression. Digistain is actually measuring what is happening at the coalface in the cells. And it's a hallmark of many solid tumours, where we've been approved for a national clinical trial here in the UK for multiple tumour types, including kidney, bladder, colon, and prostate and lung as well. So in the development pipeline, we have a few other indications coming very, very soon. Thanks very much. Can we take some questions, please?
Thank you, Dr. Amrania. We don't seem to have questions at this point in time. I think everyone's digesting the information that you provided. So I'm happy to end this session. I want to thank everyone for attending the DigiStain webinar. And we hope that you found the presentations by Professor Charles Coombes and Dr. Himal Amrania both enlightening and valuable. A special thank you to our esteemed speakers for sharing their expertise and groundbreaking research with us this evening. Your insights are invaluable and greatly appreciated. And your passion and commitment to advancing healthcare are truly appreciated. Together, we can continue to make strides in improving patient outcomes, and we look forward to seeing our attendees in our future events. Thank you very much.
Thank you all.